So looking forward to the half day, looking forward to learning more. And now it's a great, great pleasure. I think you're really going to enjoy our keynote speaker, Dr. Lindsay Harris from the National Institute of Health. I'll very briefly give you a few background points about Dr. Harris. She's a medical oncologist. She joined the National Cancer Institute in 2017, and she was before that for 30 years a breast cancer research and translational scientist. At NCI, she is really in charge of the biomarkers. And as you know, biomarkers are what we hope integrated diagnostics will give, will be data-driven, multimodal, predictive, and prognostic biomarkers. With this in mind, Dr. Harris, would you please start? And thank you very much for joining us. I know your schedule is unbelievable, and we understand that you cannot be here in person. Dr. Harris.
Well, thank you very much. I'm just bringing up my slides. Can you see my slides?
Yes, we can.
Okay, great. Well, thank you, Hedy, and I'd like to thank the organizers for inviting me to represent the National Cancer Institute today. What I'd like to do today is to describe to you how NCI has helped to support the development of precision medicine and integrated diagnostics over the last decade. I have no conflicts of interest. So let me start with MATCH. NCI MATCH is a precision medicine trial that explores treating patients based on the molecular profiles of their tumors. It's for adults with solid tumors, including lymphomas and myelomas. And these are in patients whose tumors are refractory to therapy or for whom no therapy exists, no standard therapy exists. So how does MATCH work? Well, patients donate a biopsy, and that biopsy is sent to a central laboratory where targeted panel sequencing is performed. Tumor testing is then evaluated by an AI expert system, and if a tumor gene abnormality matches to a treatment arm, the patient is assigned to treatment. But how does it actually work? Well, the patients were asked to donate a biopsy by sending out a standardized kit to the physician's office, and the biopsy was dropped into formalin and sent to the central pathology laboratory at MD Anderson. There, the tissue was accessioned and processed, and nucleic acid extracted, as well as immunohistochemistry for various markers performed. The nucleic acid was sent to one of 4 central laboratories to perform a targeted panel, and these contained over 300 cancer genes and various variants, including fusions. These laboratories were harmonized so that they were all performing the same test. The result was then sent to the Ion Reporter, which identified mutations of interest, which were then fed into the Matchbox. Now, the Matchbox is an AI-driven system, what's called an expert system, and this integrated the genomic information, the patient's personal information, including their histology. While MATCH was a tumor agnostic study, the histology was necessary because some histologies were excluded for treatments that were already standard of care under those histologies. The result was then sent out to a central review committee that deliberated on the result. And if the patient was identified to have an actual mutation of interest that matched one of the treatment arms, report was sent out to the clinical database as well as to the clinical record. and the patient was matched to therapy. Well, sometimes there was a situation where there was more than one AMOI. So if we had tiebreakers, we used various features to determine which won the tie, and that included the levels of evidence, the variant allele frequency, and things like which arm had accrued more to the study. So when we began MATCH, as I mentioned, we were having fresh biopsies sent from the patient and nucleic acid extractages and central assays performed for using a targeted panel. The result then went on to a central review committee. Now, I should mention that the review committee consisted of medical oncologists, pathologists, surgeons, and molecular biologists, as well as bioinformatics people. And they all reviewed the result to determine whether it seemed reasonable. In fact, the number of times we actually overturned one of the Matchbox results was very rare, less than 2%. Those results were then sent out in a clinical report, and the result, and the patient was assigned to a treatment arm. Now, MATCH was open in over 1,100 sites through the NCTN network. And because we had such a rapid accrual, some weeks there was up to 30 cases a day being accrued. We were able to recruit over 6,000 patients in less than 2 years. As you can see, despite the large number, we were able to perform central assays on all of them using the AI system, determine if there was a match to therapy. Over 6,000 patients were registered to the study, with over nearly 6,000 samples submitted. The number who had complete testing were over 5,500, and the number that were actually assigned to treatment based on a match was 992. Just over 600 were actually assigned to, were actually treated on therapy due to eligibility criteria from the specific arm that might have excluded them, as well as patient decision and physician decision about whether the therapy was acceptable to them. Now, after the first 6,000 was recruited, we found that there were a number of treatment arms that hadn't completed accrual. And so we decided to use a different approach to screening. The designated laboratory network was born, and that was based on the fact that next generation sequencing had become widely available in the community as a standard of care test in many sites. And so we recruited both commercial and academic laboratories to send patients, to refer patients to MATCH. And this was performed by them referring information to the physician that the patient might be eligible for the MATCH study. They then would decide whether to register the patient. And if they were registered, the designated laboratory was asked to send the variant data on that patient to the Matchbox. Again, the Matchbox would determine if there was an actionable mutation of interest that matched an arm. And a central review was performed. Again, very few were actually overturned. But in that case, the only thing that differed with the designated laboratory approach was that we did confirmatory sequencing in compared to the central assay. And the reason for that was to make sure that the results could be comparable, as we had performed the first 6,000 with the central assay. Now, these are the first 13 out of 27 reported sub-studies from the MATCH trial, which represents about 71% of all of the activated arms. There was a total of 38 arms on the trial. And as you can see, some of them met their primary endpoint, although I would have to say the majority of them did not. We continue to perform a number of studies post hoc, and those include relative studies such as this radiomic, pathomic, and genomic features in NCI match by Dr. Contos and Saltz. We were able to collect over 10,000 images through the Akron radiology group, which is associated or affiliated with the ECOG Akron cooperative group. And these images are being further evaluated and compared with the collected digital images, as well as the genomic data to determine what is the optimal biomarkers. So as you can see, we're doing integrated diagnostics post hoc as well. We're also interested in health disparities and looking at a number of disparity correlates that were collected on the MATCH study, such as the payment method, the enrolling institution type. I should mention that over 60% of the patients accrued to the MATCH study were from community sites. As you see, even despite the fact that we had the community accruing, we still were able to perform these central assays. Looking at distance from home to clinical site, looking at other neighborhood-based features such as metrics reflecting structural racism and discrimination, socioeconomic status, indices, and rurality. The kind of questions we're asking is what is the distribution of disparity correlates across the NCI match study and how they associate with one another, sex, age, race, and ethnicity. Also, are the patient's clinical condition associated with disparity correlates, including the metastatic site and treatment history? Do potentially modifiable risk factors such as BMI or smoking different from disparity correlates? And are tumor molecular characteristics associated with disparity correlates? This work is also ongoing. Lisa McShane leads it, and we hope to have some results fairly soon. Now, we haven't stopped there. NCI continues to develop precision medicine trials, and we have 3 new large precision medicine trials, including Combo Match, which looks at combinations of agents determined by genomic testing, MyeloMatch, which is looking at acute myelogenous leukemia and myelodysplasia and matching them to therapy, iMatch, which is looking at dividing patients based on tumor molecular features into various buckets and treating them with immunotherapy. And the Combo Match will still use the designated laboratory network, which was developed in MATCH. In addition, MyeloMatch will use the and iMatch will use the molecular diagnostic network, which is NCI supported and will supports the biopsies as well as all of the molecular assays performed on the trial. And then finally, the PMAC, Precision Medicine Analysis and Coordination Center, is our updated and refined matchbox. And just to detail that a little bit more, the further developments include sequencing pipeline configuration, multi-assay ingestion and integration, seamless integration with laboratory and clinical systems, biospecimen tracking, parsing, annotating, and molecular reporting, automated patient management workflows, treatment protocol management and tracking, algorithm-driven treatment assignment, data analytics, visualization and reporting, protocol development support, and actionable alteration identification, review, and curation, as well as NCI designated laboratory network support. So as you can see, we've really come a long way. This is the design of the IMAC study, where patients who are receiving immunotherapy will be divided into buckets based on their tumor molecular burden and their tumor immune score by the NanoString assay into 4 different buckets and treated with immunotherapy. And we will determine how each of these subgroups respond to treatment. Later on, then, the different subgroups will be identified and treated with specific immunotherapy combinations based on not only their subgroups, but also molecular features. This is the MyeloMatch study, which is fairly complicated and involves a number of tiers of therapy. The first tier is for the newly diagnosed AML patient with initial treatment, which is determined by cytogenetics flow and targeted panel selection with targeted therapy. These are tier 1 treatment trials. And then the tier 2 trials are designed to evaluate patients in CR using MRD-based assignments, MRD by flow. And the third tier are trials designed to evaluate patients using MRD-based assignments, either with transplant or cellular therapy. And then the fourth tier are participants with low disease burden, designed to validate clinical utility of NGS and other assays. And specifically, we will be performing error-corrected sequencing, which is a much more sensitive method than flow, comparing that to flow. And also, we will be looking at different assays that are more sensitive to try and improve the outcome of patients. And finally, the Matchbox will, of course, be used to integrate all of this information along with the local diagnosis, the subtype of AML, as well as the age and so forth to determine whether they go in, which basket they go in, and which targeted therapy they are assigned to. So thank you very much for your attention, and happy to take any additional questions at the time of the panel.
Dr. Harris, thank you very much. Most interesting and informative, and I'm sure there will be many questions during the discussion. Thank you.